Okay, so welcome to the last talk at the workshop, and our speaker is Anna Marciniak-Czochra. She is a full professor at the University of Heidelberg and the director of the Institute of Applied Mathematics there. Her research is concentrated on mathematical modeling, in particular in microbiology, and more specifically in morphogenesis. As a tool, partial differential equations are used, reaction diffusion systems, and all this is done in collaboration with biologists. She has just got her second ERC grant for collaboration with biologists. ERC grants are very prestigious and highly competitive European grants awarded only to the best. But maybe what is more, still more prestigious, she is a co-spokesperson of the Heidelberg Cluster of Excellence. Clusters of Excellence, this is a form of organizing and financing research. And I think that there are not more than 10 of such clusters in Germany. And of course, university had to write an application and then win in a competition. And this is really a great achievement. And let me say the name of the cluster. It is a unifying approach to emergent phenomena in the physical world, mathematics, and complex data. So you can imagine how difficult it was to get together physicists, mathematicians, and computer science and make a reasonable project. So this is really a great achievement. Anna Marciniak-Czochra co-organized 30 conferences and gave 36 talks only in the last 5 years. And she promoted 11 PhD students and supervised 10 postdoc fellows. Okay, Anna, please, the Zoom is yours.
Thank you, Eva, for the introduction, which is overestimating everything what I'm doing, but very nice. Thank you. So it's my pleasure to give this lecture here, and Now, my aim in this lecture is to show you some mathematical models, model analysis, and application to the data in context of stem cell-based processes of some sort of biological processes, which I will explain. And I would like on one side to explain you modeling approach based on these examples. On the other hand, I would like to show you how newly emerging data challenge mathematical modeling and in fact immediately require us to think about new mathematical concepts and underlying analysis. So if you find anything like too fast or too and need more explanations on, especially on some modeling stuff, just please don't hesitate to interrupt me. So I will be happy to slow down on any slides, and it's like we don't need to go through everything at the end of the day. Yes. So I would like, like if we think from a generic perspective, I would like to show you some examples of mathematics of structured populations. So we can say that populations, structured populations are everywhere. So population, what means structures? It means they are population which exhibit heterogeneity, but this heterogeneity is functionally relevant. So it means it decides or rules the processes we are interested in. For example, if we have age-structured population and the processes like reproduction depend on the age, or if we may have also space structured problems, but in this lecture I will not focus on spatial structures. So the structures can be very different. Yes, can be discrete or continuous. We can speak about some certain classes and then have, for example, 5 different structures, like in all these epidemic models, if you speak about healthy, infected, recurred or resistant, then you could have few classes, and these are discrete structures. We may also speak about continuous structures like age, size, and then we, of course, can think about dynamics, which depends on multiple structures or multidimensional structures, and structures which are dynamically regulated. And such problems and then also models appear in very different fields, of course, not only in biology and medicine, but since my focus is on biology medicine, so I put here this main biological fields like epidemiology, cell developmental biology, and medicine, but in fact you can imagine them everywhere. So I would like to focus in this talk on examples of describing such heterogeneity, functional heterogeneity in case of stem cell based dynamics. And I will focus on 2 systems which are relatively simple because they don't consider the space. So generally speaking, if we think about stem cell based systems, we think about this population of stem cells. Stem cells are cells which have high capacity for self-renewal and producing different cell types. So here we can look at the example of blood production system. And in fact, the blood system was the system when a concept of stem cells was also in biology proposed for the first time. And then there is this classical understanding of the process as a tree-like structure when you have on the top stem cells, so it means cells which have a high potential for dividing, and then after division they can produce more stem cells, or they can differentiate into more mature cell types. And in some points of this tree, we may have also bifurcations to different lineages, because at the end we need white blood cells, red blood cells, immune cells, and many different cell types. And this structure, stem cell based structure of the tissue, is not only relevant for tissue development or regeneration, but it's also relevant if you study cancer, because cancer often means mutation of the cells which are pretty early in these lineages, like stem cells or stem-like cells. And then if such a line is mutated, then they don't produce correct mature cells. They also send wrong signals, get wrong signals. Again, if we think about the blood, we think about here about leukemia. This is white blood cells cancer, and this is exactly mutation in some early stages. And then there is not enough mature cells, but at the same time, the stem cells get signals that they should produce more, and there is overpopulation of these malignant cells. So we would like to understand such systems, and just not to give an impression that all stem cell-like systems, for stem cell-like systems, space is irrelevant. I would like just to tell you that if we look at the blood, or we will also look further in this talk on neurogenesis, so neural stem cells, we just focus on tissues when or in blood, if they are in bloodstream, they are well mixed, or even when they are in bone marrow, when the stem cells are sitting and reproducing, we think about systems which are spatially statistically homogeneous. So we can neglect the space. However, of course, in development of many systems, tissues, organs, like here you have some organ plant meristem, when you have stem cells in the middle, and these stem cells, when they divide, they produce more stem cells, but also if the tissue is growing, they produce differentiated cells, and this is how the organ develops. Or even more complex, here is a fish respiratory organ, when stem cells are sitting in some niches just on the boundaries, and then produce new cells who move and then move in other directions and produce more cells. So as you see, spatial structures might be very important, but we focus on the system when we can neglect. And instead of looking on spatial structure, I will rather look in this talk on the structures related to different stages of differentiation. As I said, in this classical setting, it corresponds to this lineage tree, when I have stem cells. And then when I look at just one lineage, I have stem cells, I have different types of progenitors and up to mature cells. And I want to describe each of such cell population using an ordinary differential equation. I will explain you in details in the next slide. Alternatively,
quick question.
Sure.
So typically, what is the size of n in such system of equations?
It is a good question from the biological point of view, because usually this states, okay, in blood, you can consider 6 or 8 already physiologically well-defined states. However, investigating the models, we showed that, in fact, if you consider 3 compartments, you can always in the model put some substates together and just do some approximations. So in case of neurogenesis, we will, just depending on the question, we'll study even 2 compartmental models just for some early subpopulations, or we will study like 6, 7 compartments.
Okay, sorry. So this n corresponds to the number of states, not to the number of cells in this whole system. Yes. So probably
yes,
it's manageable to do it analytically. I don't know. It is not like one system of 1 million equations. Yes.
No, definitely not. And I will address such questions about heterogeneity. So which heterogeneity is really there, is relevant, and how we model it, how we try to understand it with the models. So again, we focus now on the system which has many enough cells to describe it as differential equation. When I showed you, for example, this example here, there is just 5 to 9 cells sitting in the niche, and of course this process should not be described by differential equations. But here I will first focus on blood, and in blood you have 10 to the 9 cells at the end, these mature cells, and 10 to the 5 or 10 to the 6 of the cells in the early compartment. And that's why we put them to these compartments, which we assume to be well mixed. And we describe evolution of such cell subpopulation using ordinary differential equation. And then this structuring means, but I will just explain you in the next slide model in details, that I simply, for this different subpopulation, I allow different parameters for their division, their depth, or other properties. Yes? It's okay?
Yes, thank you.
Now, alternatively, one could think, on one side, if one would think, okay, maybe I don't know how many compartments I have, maybe from mathematical point of view, one could consider to have here continuum. And I will also show you on an example of a model where we in fact find out that this is not a good idea for the systems. However, continuous model may be also relevant if you simply think that coming from one state to the other state, cells don't divide, but just there is kind of continuous transition. I will come to this later. So if we now think about this multi-compartmental model, so exactly the key assumptions and key components of the model. So we assume that each of this population consists of cells which are dividing. So if cells are dividing, then I have division rate or proliferation rate, which is P in my equation. So says how often cells divide or number of division per time unit. Then I may have death rate, which in fact for my lineage I will now consider only for the mature cells for which we know this turnover. We know what is the lifetime of white blood cells, and then we can even quantify this parameter. But we can also consider death rates for all intermediate compartments. And then we have also one very important parameter, which we call self-renewal, and which should be understood on the level of the whole population, which says that how many daughter cells are the same as the mother cell. And you know, the division goes through symmetric or asymmetric division. So it means if it's asymmetric division, it means that stem cells, or when it divides, it produces one stem cell and one more differentiated cells, or it produces then it's symmetric if it produces two cells which are the same, and it could be two differentiated or two stem cells. And so if you look at the level of the whole population, the fraction of this daughter cells which are the same as the mother cell is our fraction of self-renewal. And this could have also this interpretation on the level of probabilities for the daughter cell to be the same as the mother cell. So naturally, this parameter is between 0 and 1. So then what is the dynamics? If we look at any of the dividing cells, like all without mature cells or except mature cells, you say that the cells of I type divide at the rate P, and then two daughter cells with this probability AI stay the same. There is -1 because mother cell does not exist anymore after division. So this is the self-renewal, the repopulation. Yes, this is what is the growth. And then you have for each compartment, of course, except the first one, you can think about the flux which came from the previous compartments. So it is just those cells which were produced in the previous compartments, but just those who went to the next one are with the probability at the fraction 1 minus a I minus 1. And again, two daughter cells. So you have then this description of the model. So if you look, of course, this is except of the last compartment because they don't divide. So they will only have this transition from the previous, and the previous there is no flux from anybody else. They are just having this growth. And each of them, especially the last one, can have also death rate, which is just a linear decatur. So if you look at such equation, it's definitely a system of linear ODEs. So the dynamics will be exponential growth on exponential decay, unless we assume that all fractions of self-renewal are 1/2, and then the system would stay in an equilibrium. But if you look at the data, so first of all, if you look at the system, even for aging systems, blood counts stay more or less stable. So it means the system is in homeostasis, is in equilibrium. So it's definitely there might be regulatory feedbacks. And how these regulatory feedbacks work, it is also not completely understood, but one of the regulation is known through signaling molecules, cytokines in case of blood, and these cytokines are produced by surrounding tissue, then undergo natural decay and can used up by some cells from the bloodstream. For simplicity, we may assume just from mature cells. And now if I have such an extra ODE for signaling factor, I can even rigorously go through this to a functional dependence through a quasi-steady state approximation. So it means we assume that the signaling process is much faster than division. We put the derivative to 0. We have an algebraic equation. And from this algebraic equation, we compute a signal. And in fact, here I already rescale it to signal intensity just by rescaling the signal so that we have how the amount of mature cells influence this signal. And as you see, this is a Hill function, which is often used for nonlinear regulations. Okay, so next question is which parameters are regulated? In this model, we don't have much choices. It can be or proliferation or self-renewal. And one could even think proliferation would be pretty natural, because if you want to regenerate faster, you just push cells of early stages to divide faster. Yes, so it means if there are not enough mature cells, proliferation growth. Alternative mechanism would be if there is not enough of mature cells, self-renewal growth, what means at the early stages, stem cells reproduce more themselves and then only then differentiate. And if you feed this model to data, we find out that this is indeed the self-renewal parameter which must be regulated. So now what are the data? We are here very lucky because, as I told you, the system normally is in homeostasis, so pretty boring data, but we have data on the response to big perturbations. And these are data from clinics, from patients. And in this plot, you see data for quite a few patients. And these are data after chemotherapy, when this white blood cell counts went down, the patients got a transplantation of bone marrow, which have the cells which are considered to be stem cells, but also early progenitors. And then within 2-3 weeks, there is a regeneration. And in fact, this is a very critical clinical issue because this is the time when the immune system is not working properly and the patient can get sepsis. So in fact, this was the very first question which we got from hematologists when we started our modeling work was which aspects of the system decide about this time of hematopoietic reconstitution. And when you feed the model to this data, model with regulation of proliferation, the blue line of regulation of self-renewal, red line, and regulation of both black line, then you see that in fact regulation of proliferation would be a very inefficient mechanism. You would need 1 year to get repopulation of the cells. While regulation of self-renewal is very efficient, and of course, if you regulate both, it will be even better, but only slightly better. So this showed that regulation of self-renewal fraction is the most effective mechanism of hematopoietic reconstitution, and that this is in fact sufficient to explain the data. We also tried it here with this different types of reconstitution for the patient, also with clinical data, and in fact, the model could reproduce all of them in a very good way. Okay, so now
question: what does
Yes.
it mean in practice? Regulation of self-renewal fractions.
Okay, yes, please. Yeah, no, it is of course. I mean, what self-renewal is in this model? It is the proportion of symmetric self-renewing divisions versus all divisions, more or less, yes? So the more stem cells reproduce themselves, the higher self-renewal. Or the more there is more differentiation, this self-renewal is slower. And this is, of course, a very difficult parameter to catch it biologically, experimentally. You cannot measure it really or you cannot influence this. I will come later to this importance of this parameter for cancer dynamics. And unfortunately, it's not that we can easily manipulate it, but it improves a lot understanding of the system, and I mean, there one may search for ideas how to measure it, or how to manipulate it. Now, maybe I will just tell you, I will not show you here computations, but I will tell you, can we learn here anything from the analysis? And already as somebody of you said that, okay, if this model has not too big n, or even in case you know the structure of equation is pretty nice, you can get also some results for general n. We can use very simple analysis of the ODE system. And I put it now together because it will help us understanding of more complex cases. First of all, we have always a trivial steady state, but this is always unstable unless it is the only equilibrium. Then interestingly, we can have semi-trivial steady states. So it means that some early stages could die out. If self-renewal of one certain stage is the highest, then it would take on the function of stem cells. And then we have just unique positive steady state, which is locally asymptotically stable. This we can do with linearization. And for example, in case of two compartments, we can also prove that it's globally stable using this already for two compartments pretty fancy Lyapunov function. And if you have more than two compartments, we can also find a regime of Hopf bifurcation. So it means oscillations may emerge. But it's interesting that these oscillations emerge for parameters which are very far away from the parameters estimated for healthy hematopoiesis. And in fact, already two parameters must be mutated. So this is the dynamics. And now what can we do with this model further? So as I said, interesting question about stem cell hierarchies is in cancer. And we can relatively easily extend the model to model acute myeloid leukemia. So when we say, okay, we have our cell healthy lineage and we have leukemic lineage. And since in blood, we know that the two coexist, we need some competition between them, or we need to decide, in other words, to decide how leukemic cells answer to these regulatory feedbacks. Okay, so one easy concept is that they answer to the same cytokines. So it means now instead of having my N equations, I have N plus M equations when I have M stages for leukemia, and then I have the same feedback. And of course, though here we usually, since we saw that 3 compartment models can reproduce system dynamics pretty well, we may consider 3+2 equations. And then again, we can do linear stability analysis, and we can find conditions under which healthy cell line gets unstable and leukemic cells growth. And interestingly, this condition it's given here. So you have leukemic in red, healthy in green, and you have some algebraic relationship. And this is interesting that usually it is believed that higher proliferation or a smaller death is the main feature of the cancer. But again, from this relationship, you find out that in fact, the most effective for cancer to win is to have higher self-renewal of cancer stem cells. And this does not, this result does not strongly depend on the choice of our regulatory mode. What is also interesting, so we may say also maybe cancer cells don't respond to the healthy signaling. Alternatively, one could think that cancer cells are not regulated, but at the end they sit in the same niche in the bone marrow together with healthy cells. So at the end, if the healthy is too crowded, the cells are kicked out from the niche by competition. So we may just model it through cell-dependent or cell state-dependent death rate. And although the underlying mechanism is very different, at the end, mathematical structure of the model is nearly the same. So we can again prove that self-renewal of cancer stem cells is the key parameter and decides about cancer development. And we may come with this observation also closer to the data. And so we would like to understand how dynamics of disease changes if parameters change. So what is dynamics of the disease? One of the important parameter of this dynamics is time to recurrence. So by acute myeloid leukemia, this is recurrence is always happening. So unfortunately, so the question is how quickly after chemotherapy there will be a new emergence of the cancer cell population, and we may observe now how relative time to emergence of such relapse depends on changes of the parameters. And we see that if we increase parameters, then of course these times go smaller in case of stem cells, cancer stem cells. So we see big differences, while if you look at the intermediate compartments, there is nearly no influence. So somehow this observation allows us to think, okay, let's forget for our modeling purposes about the intermediate compartments and approximate the whole system by 2 compartments, dividing cells versus mature cells, and for cancer, dividing cells, so cancer stem cells versus non-dividing blasts. And if we have such simplified model, then we have not many parameters. Parameters of healthy cells can be used from those which we estimated from this previous data types. So in fact, we may now use data of patients with cancer relapses to estimate cancer parameters. And then here you have such data. You see you don't have, unfortunately, too many points. So there was a patient with such a number of blasts, then chemotherapy, the numbers went to 0, or to very small, but then there was a relapse here after less than 200 days. So, and here we have even data from patients with several relapses. And then if you estimate model parameters to this data, you find out a line on which cancer stem cell parameters may lie. So you have always 2 parameters, self-renewal and proliferation. And since in our models A is always multiplied with P, then it's clear that I cannot identify them uniquely. So it is like, but I can always find this curve on which they have to lie. So I can do it for different patients. Here is the data we use data of 31 patients with relapses, and we identify their self-renewal proliferation curves. And then we look statistically on the relevance of this curve. I mean, this is purely, let's say, mathematical measurement because you cannot measure self-renewal. And then compare it with disease dynamics and survival of these patients. And this showed that, in fact, this is statistically relevant, and those patients who had these curves in this upper right corner had much worse survival than those who had parameters located here for smaller smaller value of its multiplication. So this shows that this model allowed us to estimate some measurements for prognosis of disease. And if you look at data with several relapses, you see that this curve is moving in a bad direction. So this also suggests that, in fact, with relapses, a self-renewal of cancer stem cells is increasing, what could also explain their resistance to chemotherapy, because chemotherapy is attacking cells which are fast dividing. So it is just a small example of the relevance. And now, actually, if there are any of you who are more interested in PDEs, I would like to just show you just few slides about possible extension of this model to continuous differentiation process. Especially when we started all this modeling works, one could have identified 3 trends in modeling of such processes, namely using ODEs, using transport equation like continuous model, or using delay differential equations, which are kind of transport process like continuous process. And usually one have a feeling that this decision which system one uses depends on which are her favorite differential equations. So we wanted to understand in how far this process, which we so nicely described with ODEs, could be also described with PDEs. So assuming that we don't know how many of these intermediate stages, assuming that they are many, and assuming now that we can approximate them by a continuum. So if we just rewrite the model I wrote previously under these assumptions, I get an ODE just the same as previously for U1. So this is now ODE for the stem cells, which in fact could be also here glued together with the second equation. Then I get equation for this continuous differentiation, which is transport equation, first order hyperbolic equation. And then you have just this continuous transport with some velocity, which depends also on cell states. And if you want to have really equivalent, continuous equivalent of the discrete model, we can, we can just write it explicitly, and then you have proliferation and death. And then the last compartment is describing mature cells, and here we have to put it an extra ODE, because otherwise I would have some process for the continuum, which is just concentrated in a point. So if I use classical PDE approach, I could not do it. So what is the dynamics of this model? We did it just by, so if you discretize this model, you get back to the ODE. So one would expect actually the same dynamics. And this is first interesting observation from a mathematical point of view, that the dynamics is not necessarily the same, namely already structure of steady states is different. Now I can have, of course, trivial steady state, but I can have also strictly positive steady state. You know, you can compute here steady states, again, putting just time derivative to 0. And the only difference is that you would have here this first order equation with respect to x to solve it. So you get the equation just depending, just the solution of this equation. So here just for the 2 points. So I didn't put it for some reasons on the slide. So, but the dynamics is more like multi-compartmental model, because you can still have hop bifurcation and all this complex dynamics. So this already shows that the models are not really equivalent unless we assume some specific range of parameters. And this can be also seen more rigorously if you try to get continuous model from a discrete one just in a limit. So if we departure from the discrete model, so now to go to the continuous limit, we need to assume that the distances between compartments tend to 0. And if we assume that one go from one to another compartment through division, it means we have to assume that proliferation rates tend to 0. So this might be a good idea if you would consider evolution over, I don't know, millions of years, but definitely not reconstitution in 2 or 3 weeks. And additionally, if you do the limit, you see that in fact the transport appears as a first order corrective term, and you see immediately that the models, unless they are in a very specific range of parameters, are not really equivalent, and that in fact continuous description would rather make sense if we have really continuous biological transitions and you don't assume that there are proliferations on the way. And now I would like to switch to a different system, since I think it's interesting also to compare modeling and to see that how important is for the modeling is to have biological questions or hypotheses that drive our modeling approach. And I also would like to show you this model of neural stem cell dynamics, because it is the simplest model I have ever built, we have ever built, with probably the highest impact we had on experimental research directly. So it's about modeling of adult neurogenesis. So it means also process of stem cell divisions and differentiation, but in case of neural stem cells. And we here focus on data from mice and on 2 niches like subventricular zone where olfactoric cells are produced, and or hippocampus, and both in the adult mouse. And here it's the same concept, that there are stem cells which divide, and after division they produce back stem cells on differentiate to early progenitors, and then these progenitors differentiate to neuroblast and then to neurons. There might be also some transitions to glia cells and so on. Okay, so now we departure from a question. Namely, there are data from mice which already show immediately that the process is very different than the process of blood reconstitution. Namely, you have here data in three time points for 2 months old, 7 months old, and 22 months old mice, counting neural stem cells, active neural stem cells. So it means those who are cycling, dividing, all these progenitor cells, and you see that the process is not in equilibrium. So there is very strong decline. And okay, first of all, I would like just to comment here that these different time points are of course coming from different mice, because mouse is killed to get measurement. On the other hand, the mice are genetically the same, and it is assumed, you see here, these black dots are measurements from different mice, also for single time points, and this assumed that we can consider it as the consistent process of one system. And of course, it is considered as the problem of neurogenesis, that there is no so good renewal, and the question of aging, like, can we do anything to improve neurogenesis in aging, with this belief that it may help in some diseases and so on. So now the question is, can we understand why we have this decline in terms of system properties? And we write the model again. So first of all, what is the difference to what we did with blood? Now here we consider separately quiescent cells and active cells. Why? Because I have measurements just for active cells. So now we have cells which are just sitting there. And then if they are activated, they come to the cell cycle. We can measure them separately. And then after division, again, self-renewal means that they stay here as stem cells, and differentiation means that they go to progenitors. So I have this quiescent cells, rate of activation R, and then here B is the self-renewal fraction. So I have 2 daughter cells that stay stem cells, like quiescent stem cells, and then I have this flux feather. So here I even wrote 2 equations just for stem cells, because of course we can write also for progenitors and so far, but at the very beginning we even don't know if there is any regulation. So the idea is, look, there is this strong decay, maybe it's exponential decay, simply there is no regulation and everything is just used up. But if we apply this model to the data, we see immediately that in fact this is not exponential decay, that parameters must be changing in time. So here you have number of neural stem cells, and here you see additional type of data which we have. This is a fraction of active stem cells over all stem cells. So A over A plus Q. And you see here we have also decline. So if the process would be linear, we would have, first of all, constant fractions, and we would have also exponential decline. Here it is plotted in a logarithmic scale. So the question is, which parameter is regulated and how? And then we tried 3 different, we have 3 parameters, we can try 3 different scenarios. One is regulation of proliferation, saying that proliferation is coming down, but in fact the model has the same type of dynamics. Second one is regulation of activation, saying that activation is smaller with aging, so it means old stem cells are more lazy. And this fits very well to the data. You have also here this decline. And the third hypothesis was increase of self-renewal. You get less differentiation, so it means maybe you just have more self-renewal of stem cells. So we just use this kind of curve for self-renewal. And this also describes saturation of the decline, but numerically, like comparing different criteria for fit of the model, you find out that the regulation of activation is better. You can also compare the models in this way, that you have look at a model of regulation of self-renewal, regulation of quiescence, and then regulation of both. And in fact, if you allow system to regulate both, the best fit is very similar to this fit of regulation of quiescence. You can look at the estimated parameters. If I allow that both parameters are time dependent, the best fit has nearly constant self-renewal and just regulated activation. So that's why we concluded that during aging, stem cells become more quiescent. And this has a very nice follow-up in the experiment, and just first of all telling to experimentalists search which aspects of the system influence activation and then change with aging. So interesting was that typical cell cycle properties were not changing in aging. Here you have plots, measurements for young and old mice. But this is also what model predicted. And then we also look at the distribution. So in this picture, you see distribution of the cells old and young mouse along the lineages. And then you see also that there is more quiescence for the old mouse. So the question is, what causes this regulation? And then one of the natural ideas, maybe it's related to the inflammation process. With aging, all our tissues undergo inflammation. So because then the idea is, what happens if we would make inflammation, make inflammation smaller? And just to investigate the role of inflammation, we did interferon knockout experiments. So these are experiments which are kicking out response to inflammation. In some sense, you can think like this. And then you can compare the data, black dots, of the wild type, total stem cells and fraction of active, and these experiments. And then if one thinks it would stop decreasing activation, what one would expect that now we have exponential decay, but it seems not to be the case. It seems that there is more decay at the beginning, but not later. But the fractions indeed change, and they are now more constant. And if you feed the model to the data, to the both type of data, what is very nice result is that the model now predicts that activation is not regulated anymore. So somehow the model, the model feed find out this property of the system after this knockout of interferon. But the model also explains that the not big change for long times is due to the fact that now self-renewal got regulated. So that the system has a kind of compensation for this loss of activation regulation. So another question is now how these processes are really regulated, because so far we only use this time dependence. And if you want to understand feedbacks, then of course, what we do usually in the modeling, we just assume some feedbacks, or maybe we know some biological knowledge as previously about cytokines, and I put these feedbacks into my parameters, and then I check how model is working. So since now we departed from the model without these assumptions and found these time-dependent parameters, now I know which parameters I would like to get regulated. This is or activation or self-renewal. And since I know how they depend in time, and I know how populations depend on time, I may guess that my activation rates, and then of course I can show it by model fits and so on, that my activation rates must be parameters which have this kind of Menton-Michaelis type dependence on the cells, while self-renewal has this type of Hill function. So it is exactly as we had it also in the blood, but now we can get it directly from this model and from the data. What it means? It means as previously in the models of blood, this signal for self-renewal must be produced by some other cell types, like outside our stem, like by the niche or by other organ, and then consumed by lineage cells, while the the signals for activation is produced by the cells of the lineage. And I wrote here cells because, in fact, a priori, I don't know if these are quiescent cells, active stem cells, or maybe something from downstream of the lineage, like in the blood we had here just mature cells. And if we would follow our favorite hypothesis, so maybe so that I have here as in blood, like neuroblasts, like cells which are more differentiated, and then for activation, maybe quiescent stem cells by themselves, I could get a very good fit to different type of experiments. I mean, we also extended the model to the feather compartments and then fitted the model to the data on the feather compartments. The problem is that I can put here as cells, any of the cells, and I will have the same fits. And this is also, I think, interesting to see it, because it shows how important it is that we not oversell the results. Because if we would come with one hypothesis, we would prove it, let's say, by model fitting, and could say that we know the mechanism. But in fact, from this data which we have, we cannot distinguish the mechanism. And now the question is, do we have other type of data, like maybe big perturbations, which could distinguish the mechanism? Or do we have more biological insights, which could tell us what are exactly molecular signals which are doing it? And then we would know which cells are producing it or which cells are taking it from the system. And then at the end, I think it's interesting if we compare the two systems, and this is again advantage of modeling. Otherwise, how can you come to comparing blood with neural stem cell systems? It is that we see that the most significant difference is homeostasis, equilibrium in blood, versus decline in the neural stem cell system. And in fact, this can be all explained in terms of the self-renewal parameter, which in blood models is larger than 1/2, and in the neurogenesis must be smaller than 1/2. And even more interesting is that if you go to less to a system which is less developed in the evolution than mouse, is like a fish, then you can also observe that neural stem cell system can be also in equilibrium. So it's somehow that this everything depends on this self-renewal. And of course, the question is, how can we interpret it and study it further using experimental data? And I have still 5 minutes. Yes, Eva?
A little bit more, because there was some, we started a little bit later. So,
because actually I would like at least shortly to show you, because somehow I showed you models which are, at least for biologists, even for clinicians, pretty convincing, helpful, even could help experiments, but from mathematical point of view, they are also relatively simple. Although, of course, we have also versions which are maybe more involved, but let's say basic ideas are relatively simple. But I would like to show you a new type of data which immediately bring us outside our usual mathematical tools. These are data which are now celebrated in cell and developmental biology, also in oncology. They are so-called single cell data. In the single cell data, information is obtained for single cells. This is using single cell sequencing. And then, okay, we deal with data about transcriptomics, about mRNAs. But they are also this type of data for genetics, for epigenetics, for metabolomics. So different properties of the cells are measured on the level of single cells. And then such cells are then quantified in high dimensional space. It could be like 500 dimensional space or more. And now the question is, what can one do with such data? So far, the data are mostly treated by bioinformaticians and are treated in this way that, just using some linear algebra. First, the data are, the high dimension is reduced to a relatively small dimension, maybe first 20 or something like this, just using principal component analysis, so just finding the direction in which you have the biggest spread, like eigenvalues, eigenvectors, and so on, and then finding out some main components or linear combinations which would define as this principal components. And then there are different approaches how to bring this to pictures which are two-dimensional, which biologists would like to have to see. So they are fancy approaches like this TSNE, from which we have this data, and these are our neurogenesis data, is, for example, done using some fuzzy topology. I mean, I cannot explain you this more, but maybe more used approach is so-called UMAP, and in this approach, there are some physical model applied to the data, which define some kernel of attraction for individual cells in the space. So the cells which are close would come more together, the others would be repulsed, more or less, and then you observe like kind of better spread of this data in these spaces. And these are the data usually which biologists use for their, let's say, interpretations, saying, and here if you assign on this data which cells are stem cells or neuroblasts or active stem cells or neurons, or maybe not neurons, I don't know, because you see here different cell types for different colors, you may think now, okay, in fact, they align like in our interpretation that you go from quiescent stem cells to active and then to some progenitors, neuroblasts, and so on. But somehow these are pictures. And in fact, if we assume, and this is assumed that everything is in Rn, in Euclidean space, then even we know that we don't have this, that these are not real embeddings like this two-dimensional embeddings. They are some kind of simplifications on the way and reductions. So are this data useful? And then and how to bring this data together with our models. So somehow this was the task which we obtained from our experimentalists. And if you think that that you have here the clusters, you could say, okay, so it means these blue guys are my Q, then I have my A and other compartments from the model. But in fact, you can forget about this because first point is this is just one time point. And second point is that if you look carefully at the distribution, like if you even try to define one-dimensional direction of this lineage, this is doing through finding kind of average curve here. And you project all this distribution along this line. This line is called pseudo-time because it's kind of resembles direction of time evolution. And you see the distribution of different markers, so different cell types along this line, you see that there is nothing such as real clustering. I mean, this clustering is just picture. And indeed, you cannot say that quiescence are only this blue or something like this. So there is a problem of the structure of the cell state space. Can we do it one-dimensional? Can we assume this kind of clustering? Also, it's clear the process is not linear. We showed it previously, but you also see it here. If you look at the distribution for young and old mouse, you see the distribution changes, so it cannot be just a continuous transport growth process, which is linear. So the question is what to do with this. And we think that here one could use a pretty generic approach to structure population model. And in fact, one should use its ability to describe processes in a pretty generic spaces. So this is a very generic structured population model. So because you have time dynamics consists of continuous transport, of growth decay process, and of a kind of transitions which are just defined by some kernel. So here you could also describe, for example, a kind of random dispersion. And this generic model we can describe in the spaces of non-negative radon measures. And now I will skip maybe this because we don't have time, just to tell you that we can show well-posedness of such models in a pretty generic space. And this way we could put different discrete continuous models or models with just a randomness generating as this cloud to one mathematical setting. And this, why I'm speaking about this, because I would like to show you that in fact, if one looks at the real data, it's very often that the classical approaches are challenged and we need more mathematics, because now of course is the question how we analyze this model. Now so far we have just well-posedness, and the biggest problem is of course to define what is the state space. So far in all this bioinformatics type analysis, it's assumed that this is in Rn, but can we now reduce it to just one R1 and consider distributions along this space? Or should we use some graph type structure, which is which can be, for example, obtained by topological data analysis of this data? Okay, so here I will show you that also geometry can show us nice presentation of this data. So this is Poincaré map. So using hyperbolic geometry, when we can really do two-dimensional embeddings. But at the moment, we can just apply different methods. We see pictures. We can use pictures for nice biological interpretations, maybe, but we cannot use them to really get insights as long as we don't have a kind of proof of concept that this description provide us more data. And this shows also that different fields of mathematics must come together in such applications. And okay, so just an example, we put first such model assuming that the blue cloud is continuous process, and then you have just active cells, which is 1 point, and then you can write combination of this continuous and discrete dynamics now and in just consistent setting, because it's not a problem that now we have here continuous distribution versus a single Dirac delta. Maybe the last thing which I wanted to show you, because this is kind of arguing that in applications we need to bring together these different fields of mathematics. So here this was an idea coming from the fact that there are quite a few people in Heidelberg who are working on topological data analysis, and then I thought, what's about looking what this could give us for our neural stem cell systems. And what is the advantage of this topological data analysis is that if you would have data with loops and you would use this classical bioinformatics UMaps and then just reduce it to one dimension, you don't see this loop anymore. While doing topological analysis, you look at this neighborhoods of some points, and then you see when there is this kind of bifurcation. So you would have the visualization using such a graph. And if we look at our data now, these are data of healthy neurogenesis, you see a very nice structure. And in fact, with UMAP, you see a similar structure. But already when you see at the data of neural stem cell cancer, like glioblastoma in this space, you see that UMAP is completely losing this kind of loops. And the topological data analysis not only provides you more structure, but also shows you that in fact different glioblastoma from different patients can have very different structure. So maybe these results of topological data analysis could be used as kind of marker for disease dynamics and so on. But of course, again, these are just first observations, like pictures, and one now needs to prove then these loops have really meaning concerning process and concerning disease dynamics. So now in summary, I wanted to show you some example of the stem cell-based system. So for me, they are good examples how to model heterogeneous systems. Mathematical models allow to link this heterogeneity to system control, which can be nonlinear, can be also non-local. Since we now also compare these two systems somehow, and since we also see all these problems arising from the single cell data, I think that one of the generic conclusions from my work is that the choice of mathematical description should be tailored to the type of data and the underlying processes and to the type of questions. So it is significant for mathematical modeling is to have well-defined biological questions. Linking this new type of data with mechanistic models is still completely an open problem if we want to do it in an honest way so that we really can get new insights, and clearly requires integration of different mathematical concepts and frameworks. And I would like to acknowledge different groups of my collaborators. I showed you different projects, and especially my former and current PhD students working on both systems, collaborators from neurobiology, German Cancer Research Center, from hematology, from Heidelberg Medical Clinic, also neurobiology in London, in the Francis Crick Institute, and in the analysis in Warsaw. And also, if any of you would be more interested in this measure differential equation and measure settings, so I would like to also acknowledge the collaborators, and I put here, but somehow disappeared. 2 years ago, we published a monograph book on this, like on, let's say, development of functional analysis, which is needed for well-posedness of such problems. So thank you very much.
Thank you very much.
Away so that I can see you.
Okay, please. There are questions, okay, please.
Thank you for your talk. It was really interesting. And I wanted to ask, because you mentioned the influence of inflammation, that somehow maybe inflammation can cause that cells are being lazy. Could you say something more about it?
You mean from biological point of view?
Yes.
Yeah, so okay, the direct link is not now well understood. So what we managed to show that if we knock out this interferon response, so then this activation of stem cells is not changed. Yes. So now, of course, is the question of searching for connections to molecular signaling. But I mean, again, there are more speculations than real now molecular link of this parameter. So we have, for example, observations that in the aging cells you have bigger expression of SFRP5, which is inhibitor of non-canonical Wnt signaling. Yes, so one can think now about the involvement of some specific molecular signaling, Wnt signaling, which is relevant for many developmental, regeneration, and cancer processes, and just thinking how the Wnt signaling influence these processes. And of course, this is one question. The second question is how inflammation is influencing it. So everything is now just work in progress and not answer questions. For me, it's also interesting to see that the model predicts that then this increased inflammation would mean, no, not increased, so that, sorry, that decreased inflammation would mean increased self-renewal, because there is also evidence in blood that if you have more inflammation, it seems that self-renewal goes down. But again, we saw with the model, we detected correlation, and definitely we cannot say that there is causality. So it means maybe it was interferon knockout what increased self-renewal, but maybe interferon knockout only stopped regulation of activation. And then since population counts were dropping down too fast, the system had another regulatory loop which increased self-renewal. So this we don't know, yes, because the model could only detect this kind of correlations.
Okay, thank you.
Any other questions or comments? Please come closer, take the mic.
Hi. So, I mean, you used the terminology transport a few times. So I was just wondering if there's any connection between this functional analysis of radon measures and optimal transport.
Yes. Yes, so I mean, I use transport not because of the optimal transport. I use transport because for me, all these transitions, when I have my, do you see the slides? When I have my my equation, yes, this is this continuous transport. And you see what I have here in the very generic model. I allow for, so it's like, imagine about the distribution. Let's imagine that first we put some distribution to quiescent cells. So this would tell us how it is transported in the cell state space, whatever it is. Then we may have here growth and decline, and we may have here also all this kind of mixing, I don't know, kind of mutations, or this operator can describe some extra dispersion, yes, like kind of diffusion. So, and now if you think about comparing to optimal transport, yes. So if I look, okay, so maybe even on the level of this PD, so you know, optimal transport is really discussed on the level of the PDs. That's why I thought that the best would be to describe on the level of evolution of the measure. So my models, like the structured population models, can be rewritten in the form of such integral equation. This is just, if you think about classical setting, just you integrate along the characteristics, and then you find the solution here. Yes, like of course there is this is still mu dependent, so it's not explicit solution, but you find this kind of integral equation. So then the transport, instead of, I could put it on a metric space. That means instead of just transport through this differential operator dx, I can also have this kind of here transport like in optimal transport. And if you compare it now to optimal transport would have just this first piece of my model, because in optimal transport you just take some distribution and you transport it in some space. You don't have growth, you don't have decline, you don't have all this extra functions. Yes, so this would be this description in terms of optimal transport. And just to tell you this, there are also some attempts to describe the single cell data and to study it using optimal transport, where one tries to, let's say, divide, like take this distribution only of my quiescent stem cell and just infer optimal transport. So it means this kind of push forward or pull back, depends, different people call it differently, operator from the data. But again, in our system, our process is not conserved. We have also growth, decline. So in fact, the structured population model, I would say you can understand them as kind of generalization of optimal transport.
Oh, thanks a lot. It's very interesting.
Okay. Any other questions? Yes, maybe the one last question. Oh, two questions. Okay, but okay. Well,
Okay, so please go on.
On the last slide, you mentioned models, including that are not homogeneous. And as far as I understood, the first model that was analyzed, it was homogeneous. Their parameters there were time and the position up to n in this sequence from stem cells to normal cells. So may I ask, could you explain if that model concerns only homogeneous situation, or?
I'm not sure if I understood your question, I must admit. So you mean homogeneous?
For example, in space, when one consider.
In space, of course. I mean, so far, all the models I showed you are homogeneous. I mean, I don't consider space. We can say they are homogeneous in space, but I even don't consider initial distributions. And of course, if you look at this, I don't know if you ask about this structured population model. Of course, if I write it like this, it does not consider physical space. And you know, one could say, okay, let's put all these densities also in physical space. Because all these spaces which you see in the single cell pictures, these are not physical space, yes? These are the chosen two direction of all this high dimensional features. Like you can imagine there are two most important transcription factors, although of course it's not so simple. They are usually, these directions are linear combination of many of this directions. So this is not in physical space. But of course, all this you could put in physical space. In fact, nowadays, bioinformaticians are developing a lot of tools or new tools for spatial transcriptomics. But this makes things even more complicated. So I focus here on the two systems, blood and neurogenesis, when I think neglecting the physical space is well justified.
Okay,
if you would like to put it, then I think, I mean, there are many new challenges just to put spatially, because, you know, like if cells divide, where in space are new cells? Do we model it with diffusion, or do we model with some pressure, like in solid tumors, or, you know, like let's say completely many new questions, super interesting, but new.
I ask this question because I've heard that one of the first signs of cancers and tumors is that the communication throughout space is between those cells and normal cells is diminished. So I wanted to ask if there is a possibility to see such behaviors in models that do not reflect this spatial.
Yes, but you know, one of our model is strictly, yeah, I mean, I told you this, probably I put too many different things. You know, as I said, that there was this question if there is this regulation through signaling or through the space. So these guys are sitting in bone marrow and they are competing for the space. I just assume here that, let's say, everybody can see everybody. So it is like it's that the competition is global. And then I have this kind of sums here, or in continuous model, I would have integrals. So somehow I consider competition for the space, but this is given on the level of the ODE description, like you have in logistic equation growth or in all this kind of models. And for niche, we assume it because it seems that the distribution of cells also in leukemia is in the niche is more or less statistically homogeneous. And also we have more, I mean, I didn't show you here, we have more complicated models of this niche with competition, with kicking out cells and so on. But so far we mostly considered that let's say they compete for the global space and not just locally. I think it would be more relevant in kinds of solid tumors, yes, but because maybe then it's not that everybody can communicate with everybody and they do it more locally. But again, I pretty believe that one needs develop more complicated models for, let's say, questions concerning such more complicated systems. Mm-hmm. Thank you.
Thank you.
Okay, so thank you for the very interesting talk. At the end of your talk, you mentioned that some new developments in functional analysis is needed. So could you comment on that?
Yes, sure. What I just said that okay. In fact, one of the things I can tell you about is what we have just finished. It's like when we think that our processes at the cell state space is not Euclidean space, but this is kind of graph, or it's even maybe a distribution of, it's like maybe it's even infinitely dimensional space if I consider distributions of different cell types, cells in the space of, I don't know, more complex like probability measures of things like this. Then of course we needed to extend our framework to be able to describe this structural population models on metric spaces, which are maybe not even locally compact or things like this. And it's like it's always mathematical pleasure to find to do some more sophisticated analysis, but of course I think concerning applications we have to go step by step. So now, for example, is the question about parameter estimation in such spaces and so on. But still, the main, I would say, lack of understanding, in my opinion, is to find out what is really this cell state space.
Okay, thank you.
Piotr, please.
Okay, let me ask you a somewhat perverse question, if I may. You see, my brother is an oncologist. He takes care of cancer patients. How should I argue to convince him to watch the recording of your talk?
Okay, my talk was not addressed to medical.
I know.
I would do it then differently.
Okay.
So, but I'm working more with experimental biologists than with clinicians, but I'm working also with some clinicians, and I would say that maybe the most interesting from the point of view of experimentalists and clinicians would be this very simple model of neural stem cells, where in fact they were convinced before that self-renewal, which is changing with aging, and we showed with the model this must be activation, and then they did experiments and they found that this SFRP5 differences between old and young and so on. So I would say that we could really help experimentalists to understand their data, to interpret their data, and to plan next experiments. Yes, yeah.
Okay.
Yeah, thank you so much. Yes.
Well, but please, the very last question, okay? Because dinner is waiting. Okay.
Okay, sorry. I have a question regarding the methods you were using. On the end of the talk, there were, let's say, more sophisticated mathematical methods. I have a question regarding, for example, do you use or someone in such problems, for example, purely topological dynamics or stochastic calculus, for example?
We didn't. Okay, I mean, how we touch topology, I showed you. It's very preliminary, and so this maybe with the stochastic models, of course they are very relevant, and of course there are people who are doing stochastic modeling for stem cell dynamics. I think maybe one could get more information, especially if one has small populations. I cannot. I mean, we did not apply it here. What I thought now is that maybe when we are already so generic that we can put our models on Polish spaces, then we could borrow some maybe ideas and methods from the stochastic semigroup theory, from the Fellner semigroup theory about properties, so dynamical properties like of the system. And this is what actually I would like to try, but yes, but we didn't try. It's just new ideas. So actually, I think that any new idea from different methods can be useful if you think if this can provide some information. Yeah.
Okay, thank you very much.
Thank you. Let's thank the speaker again.
Thank you.
Let me stop recording.